Hello, my name is Sarah Hidalgo, and today we're going to be discussing the journal article on gene therapy coming of age. This article goes over the history and development of gene therapy in therapeutic and clinical settings. First, we have 50, 60 years ago with the genesis of the idea of gene therapy and how the scientific community first thought we were going to be introducing exogenous DNA into patients with whatever mutations were contributing to their disease, whether that be hemophilia B, hemophilia A, whatever muscular dystrophy disease they may have, cancers, lymphomas, leukemias, etc. We then have, the retrograde viral vectors along with the adeno-associated viral vectors and viral vectors in general, in the clinical trials and the research in the 90s and the 2000s, along with the CRISPR-Cas9 technology and other nucleases like zinc finger nucleases and the TALENs nucleases, and then finally CAR therapy or chimeric antigen receptor therapy, which are then attached to things like T cells to assist with immune responses and with the pathogenesis of things like lymphomas, different cancers, etc. So first and foremost, that retrograde viral vector, or the gamma retrograde viral vectors and the adeno-associated viral vectors, were really successful. They introduced appropriate genes into patients with whatever mutations they were trying to target in the clinical trials, and the general consensus is that first and foremost, the patients had really positive responses to the therapy, and they were developing really good really good proteins in response to the gene therapy. However, this did not last long. Also, they had different mutations due to the addition of genetic information. So it was genetic addition for mutation, and this would later cause different proto-oncogenes to be created, and the introduction of different cancers, along with the immunoresponses that would come with capsid introduction into patients' systems. Whether these patients were introduced to, adeno-associated viruses in the past, they would have immunoresponses to these capsids. So with that, we have CRISPR-Cas9s, the zinc finger nucleases, the TALENs nucleases, which did introduce a more precise gene splicing where particular genes could be targeted and could be excised based off of their sequences. So they could be deleted, they could be added, they can be recombined in a very specific setting. Then there were the chimeric antigen receptor therapy, or CAR therapy, which was introduced to T9 cells, which was fairly successful at first in treating different lymphomas. However, this particular immunotherapy did cause some really serious medical side effects and potentially death in some patients. So that said, I believe that all of these therapies are great in understanding how we can produce different viral vectors and use CRISPR-Cas9s in developing gene therapies, and they can have really positive effects on patients. But I also think that we should understand the ethical concerns when it comes to editing gene splicing for different germline cells, as discussed in the article, along with the ethical implications of introducing other genes to potentially create more cancers, more diseases, which is unfortunate, but it is possible. So thank you so much for listening. Y'all have a great day.